AP 120, Chapter 3, Topics Cytoplasm and Organelles. So, returning to our lovely cartoon modeled cell, we're now going to talk about the cytoplasm. And again, the cytoplasm is all the materials located between the cell membrane and the nucleus. This includes the cytosol, the cytoskeleton, and the various organelles. So the cytosol is the aqueous solution, the watery solution that fills the cell. And when we talk about a solution, in this case, the cytosol is more like a gel, more like a toothpaste than it is a normal fluid. Now, forming throughout the cytoplasm is the cytoskeleton. It is various protein rods and tubes that help to give the cell its structure and hold things in place. And then, of course, you have the various organelles, membrane-bound and unbound structures throughout the cytoplasm that perform very specific functions. So, for instance, one organelle is the ribosomes. Ribosomes are very small. They are just protein RNA complexes. They can be found bound to the rough endoplasmic reticulum, or they can be free-floating in the cytosol. And the ribosomes are very important because they are responsible for protein synthesis. The ribosome complex, when it is formed, is able to produce proteins, all the proteins the cell needs, through a process called translation, which we'll talk about more in Chapter 4. Other important notes about the ribosome has no membrane. There's no phospholipid membrane associated with the ribosome. And the ribosome is made up of proteins and ribosomal RNA, and it comes in two subunits that, when combined, is the functional ribosome. Next up is the endoplasmic reticulum. There are actually two forms of the endoplasmic reticulum: the rough endoplasmic reticulum, which tends to be closer to the nucleus, and the smooth endoplasmic reticulum, which tends to be further away from the nucleus. Now, the rough endoplasmic reticulum is composed of flattened interconnected membrane sacs. So it's this labyrinth of phospholipid bilayer membrane forming these sacs, and the rough endoplasmic reticulum has ribosomes attached to it. So these little dots represent the ribosomes. Here you can see ribosomes attached to the rough endoplasmic reticulum. And this is why it's called rough ER, because of the ribosomes attached to it. Since ribosomes are involved, that means protein synthesis is involved. So the rough endoplasmic reticulum is involved in protein synthesis and processing, basically making proteins that are either going to be part of the cell membrane or going to be secreted by the cell. Then we have the smooth endoplasmic reticulum. It is also a labyrinth of interconnected membrane sacs, but they are more cylindrical in shape. So overall looking kind of smaller or tube-like, as it were. The smooth ER is smooth because it has no ribosomes, which means protein synthesis is not a part of it. Instead, smooth endoplasmic reticulum is involved in lipid synthesis. So it produces lipids, like phospholipids. It also involved in fat absorption and the breakdown of some drugs and toxins. And for instance, because of this breakdown of drugs and toxins function, people who are IV drug users have a lot more of the smooth endoplasmic reticulum in the cells of their liver than, say, people who do not use IV drugs. So lipid synthesis, fat absorption, breakdown of drugs and toxins. Then we have the Golgi apparatus. The Golgi apparatus is a stack of flattened membrane sacs. So they look kind of like a stack of pancakes. While the ER is all interconnected, the Golgi apparatus, those pancakes are all separate from each other. The function of the Golgi apparatus is to modify and package proteins and transport them out. So it receives proteins from the rough endoplasmic reticulum. It'll do things like adding carbohydrate molecules to those proteins, and then it'll transport them to where they're supposed to go, such as the cell membrane, outside of the cell or to form the lysosome. My mind went blank. Lysosome. So that means that the vesicles the Golgi apparatus produces are often involved in exocytosis, releasing those proteins into the surrounding environment. So here's the rough endoplasmic reticulum. It produces transport vesicles that carry the proteins it produced to the Golgi apparatus. Proteins will travel through the various levels of the Golgi apparatus, getting processed and packaged, and then shipped out via vesicles to either become part of the cell membrane or to release the proteins into the surrounding environment, exocytosis, or some of those vesicles will eventually turn into lysosomes, another organelle we'll talk about shortly. Mitochondria. Mitochondria is extremely important. Mitochondria is the powerhouse for the cell. It is where most of the ATP is produced, so most of the cellular energy the cell needs to function is produced by the mitochondria. And the mitochondria, therefore, are more common. There are more of them present in very energetically active cells. So, for instance, muscle cells have a lot of mitochondria. They use a lot of energy. Another interesting fact about the mitochondria is it is a double membrane structure. There's an internal phospholipid bilayer membrane and external outer membrane. So two membranes. Pretty wild. And then ATP, of course, what is ATP? It's adenosine triphosphate. Lysosomes. Lysosomes are big enzyme-containing sacs. They are modified vesicles that were formed by the Golgi apparatus, and they are filled with digestive enzymes that break things down, which of course makes sense as to why lysosomes exist, because we want to isolate those digestive enzymes to protect the cell from them. So some of the functions of lysosomes include reabsorbing, breaking down, recycling old organelles. So if the mitochondria stops functioning, a lysosome will fuse with it and recycle it. Another function is that when engulfed materials via, say, phagocytosis are brought into the cell, the lysosome will fuse with that vesicle, and then the digestive enzymes will break down that food structure or break down that bacteria, allowing us to use the components. And also, the lysosomes can also break down some things like cholesterol, toxins, and some drugs. Tay-Sachs disease. Tay-Sachs disease is a genetic disorder where the person with this disorder is unable to produce a functional enzyme found in lysosomes. So the person is missing one single lysosomal enzyme. It is not functioning. And this is a genetic condition where the parents are heterozygotic, so they have one defective gene and one normal gene and have no symptoms. And then when the two parents who each have one defective gene have children, 25% of their children will end up with both defective genes and will have Tay-Sachs disease. This disease is very severe. It leads to the accumulation of fatty material in the child's nerve cells. Symptoms start at 6 months, leading to paralysis, blindness, deafness over the next couple of years, and the child is usually dead by age 4. Just one gene defective leading to this deadly disease. Peroxisomes. Peroxisomes are another membrane sac that is filled with enzymes. Peroxisomes, however, have a completely different function. They are involved in breaking down lipids, so they help to break down lipids. They're also involved in neutralizing hydrogen peroxide. Turns out our cells produce hydrogen peroxide via some metabolic functions, and hydrogen peroxide is toxic to the cell, so the peroxisomes can neutralize that toxic byproduct of metabolism. And that's actually how peroxisomes get their name, from hydrogen peroxide. And finally, peroxisomes also detoxify alcohol, which means alcoholics have a lot more peroxisomes in their liver cells. Cytoskeleton. Cytoskeleton isn't quite an organelle, but it is an extremely important structure for cells. It is made up of collections of long protein rods and tubes that then spread throughout the cytoplasm. You can see them shown around here. They function to give the cell its overall shape. It can also change the shape of the cell to allow it to move by changing its shape. They anchor organelles in the cytoplasms to keep organelles in certain areas. They can be a roadway, a highway for vesicles and organelles to travel along. And they're also involved in separating chromosomes during cell division. The two main kinds of structures in the cytoskeleton, there are microfilaments. Microfilaments are thin protein rods. They are involved in cellular movement and shape. They tend to be found directly under the cell membrane and so are associated with the cell membrane. So they help to form those pseudopods during endocytosis. And then we have microtubules. Microtubules are much bigger and they actually do end up forming a hollow tube. Microtubules act as a roadway, a highway for vesicles to travel along. They're found all throughout the cytoplasm, and they're also involved in other functions such as moving chromosomes during cell division. Now, centrosome. Centrosome is an organelle. Centrosomes are made of two hollow protein cylinders called centrioles. So a centrosome has two centrioles within it. And the centrosome is important because it is where microtubules are produced. So it is the site of microtubule generation. This is extremely important for the cell to maintain its cytoskeleton. It is also important during cell division because it is the microtubules formed by the centrioles that separate the chromosomes and allow for proper cell division to occur. We have some other extensions to the cell membrane. We have cilia and flagellum, and these are both motile, movable extensions from the cell membrane. If the cell has a flagellum, it only has one. If the cell has cilia, it has many of these cilia. So cilia are shorter structures, look kind of hair-like. They're very abundant on the free surface of specific epithelial cells. So they're on the side that is exposed to an open space of some kind. And what they do is they beat, they form a wave-like movement. And that movement causes materials outside of the cell to move. Cilia are involved in moving things outside of the cell. And they're found in places like the respiratory tract and uterine tubes. Flagellum, again, is one single long structure, and its job is to move the entire cell as it beats. And there is only one human cell that possesses a flagellum, and it is sperm. Sperm's the only one. And that is it for this part of chapter 3.